welcome tonight. As usual, I'll start by expressing my gratitude as an uninvited settler on Kamloops land within Secwepemc. I'll express my gratitude for the fact that the Secwepemc have stewarded this land since time immemorial, which makes it's so much easier for us, because that's our job too, stewarding the land and enjoying the nature that we find here. So we have great gratitude to them for doing that and for sharing it with us. So this evening, this is our MSc student presentation night. So we have 5 young faces here to present to us, and I'm going to have a hard time keeping them straight because although I discovered some biology 2,300 assignments from you, Sean, on my computer.
Well done, I'm sure.
Yeah, well, they were magnificent. I retired a year and a half ago, so I don't know all these guys. It's amazing, and they don't all know me, which is also amazing. Anyway, we're going to start. We have 5 presentations this evening, all different, and they're going to present to us for a short period of time, and we're going to save our questions till the end, okay? So I know you'll have burning questions, but save them until the end, okay? So our first speaker tonight will be May Frank. I just want to say my newest granddaughter is named May, and I'm so lucky that May is such a brilliant and lovely person so here you go. May.
Hi everyone, my name is May, and Sean has nominated me to be the one to thank Nancy and Gary and everyone who's invited us and taken interest in our work. So thank you everyone for coming here to hear about our work. So I'm going to talk to you today about preyability in the interior of British Columbia. So this is one of my study sites. I worked in the traditional territories of the Eskedum, SXFN, and Williams Lake First Nation. It's about 2 hours north of here. I lived in a little place called Lac La Hache. If you don't know where that is, there's Kamloops right there, and then this is my little study area. It's in the interior Douglas fir forests of the Cariboo region. So I'm looking at prey availability. So that means the quantity and diversity of food items available to a predator. So a predator can be a lot of different things, and the predators that I'm looking at are mesocarnivores, or an animal whose diet consists of 30 to 70% meat. So that'll be things like fox, lynx, and weasels. And then the other portion of their diet is like berries, insects, roots, other things that aren't considered meat. So I looked at 3 different years of forest to look at prey availability. I looked at clear cuts, so less than 15 years since harvest, 30 to 50 years since harvest, and then over 150 years where we had these very large veteran Douglas fir trees. And then what did I catch over this summer? I did this all in the summer. So here's red squirrel, yellow pine chipmunk, the red-backed bull on the top, deer mouse on the bottom. You can see the ear tag there that I used to do mark recapture and identify all the individuals. I caught a couple weasels, surprisingly. And then can anyone tell me what this blurry blob is in the bottom there? No? So that is a flying squirrel. I actually was catching some flying squirrels in my forested plots, which surprised me. So now I'm going to talk about the mesocarnivore side of that. So I've done all my trapping and looked at small mammals, and now I'm going to look at fishers. If you've never seen a fisher, it's a mid-sized weasel that lives all through the boreal forests of North America. They have wonderful coats, and they are very hard to see. So if you've seen one, you can count yourself very lucky. Most people don't even know what it is, and they think I'm talking about fishermen or birds. So this is the fisher. This is what I'm going to look at diet in. And they are one of the only animals that actively hunts porcupine. So that's an interesting thing, and we want to know if there's actually porcupine on the landscape around my study area. So how do you study diet in an animal? Well, and you can get really cool photos like this submitted by, or given to me by Shannon Worden, another master's student, and you see this fisher's bringing a rabbit leg up a tree. You can sometimes get lucky and you can get little videos. This is a fisher mama bringing a rabbit leg up to her den. And sometimes you get nice little videos like this. It's kind of dark, but I don't know. Can anyone tell me what they think that is? Here, I'll show you again. The little ball right there? What do you think?
A fisher?
Well, yeah, that's a fisher, but the little prey item it has in its mouth If you didn't know what it was, I have no idea what it is either. So this is not a really good way to look at diet. So how am I going to look at diet? And the big answer that I came up with is I'm going to look at fisher poop. So here's 2 beautiful fisher poops. So this is my study area. So I'm taking the poop, I'm sampling it for DNA, and we're going to identify all the things within the DNA so we can get the things that we also aren't able to see because they're digested down and there's no bones left. So hopefully we'll be able to find that there's porcupine in their scat, but we might also find fishers eat fish, or they're eating a lot of grouse, or even eating a lot of toads. So you're probably wondering, how did I collect enough scat? Does anyone have any ideas how I collected enough scat? You're not allowed to answer. What do you think?
Did you follow a fisher?
Shannon, the other master student, actually did follow a fisher and collected a bunch of scat for me, but I actually employed scat detection dogs. So this here is Philson. He's a rescue with rogue detection teams down in the States, and they came up and they helped me find fisher scat, as well as we found bobcat, lynx, coyote, and fox scat. He's specifically trained for all these species, and we focused on fisher. And we were able to collect all those different species, and we collected over 349 samples in the 10 days of work we did. So that was incredible. It's an amazing team to work with. It's wonderful working with scat dogs. That's Shannon on the right there, the one who collected all the other scats for me. So overall, my project, I want to learn more about small mammal populations in the interior Douglas fir. And I also want to learn more about the habits of fishers in the interior Douglas fir and compare it to wetter, more northern populations to see the differences. And again, I want to say a big thank you to everyone who came out and helped me have a successful field season. There's even more, but I didn't take any photos of them, and as well as my sponsors and the John Prince Racer Force. So, thank you.
So this is Larissa Murdoch, and she's going to tell us about bighorn sheep.
Bighorn sheep, yay! Yeah, so again, thanks May for sort of saying thank you on all our behalf. It's really fun for us to come and talk about our projects. We talk about them all the time. We love to talk about them. So I'm Larissa. I'm going to be talking about my thesis project, which is habitat selection in bighorn sheep. So just to give everyone a bit of a background, along the Thompson River in British Columbia, the bighorn sheep population has been in decline for about a decade now. It's declined around 50%. And there are 4 subpopulations that make up that bigger population here in Kamloops. We've got the Kamloops Lake North herd, which has declined by about 60%. And then we've got the South Thompson herd, which has declined by about 30% over these past 10 years. Thankfully, we have the Battle Creek and the Kamloops Lake South, which we also call the Spatsum Creek herd, which have remained relatively constant over this past decade. And those populations are going to serve as a reference study, or a reference population for my study. So I'm going to compare those declining herds to the ones that are remaining constant. So preliminary work by government biologists suggests that poor lamb recruitment is the cause of this decline in these numbers. But there are a lot of factors that can cause poor lamb recruitment, like disease and poor nutrition, which leads to poor body conditions in the mothers, and thus the lambs can't get enough nutrition from their moms and then they die. But these have both been ruled out, which leaves predation and habitat selection. Now, I'm not going to directly look at predation, but I can look at it indirectly through habitat selection. So bighorn sheep are habitat specialists. They like to hang out in very steep, rugged terrain as it limits predator movement and gives them that nice protection. And we call that terrain escape terrain. So escape terrain is these broken terrain patches that have a slope steepness of about 60 to 100%, sort of depending on your study area. And ewes really like to hang out around that area when they have lambs. But what can happen is they might have trouble balancing nutritional requirements with predator avoidance. So they might be staying or going to find forage that's further away from these safe areas, leaving them and their lambs more vulnerable to that predation, causing poor lamb recruitment. So what I'm going to be looking at is AFUs are moving away to find better food and making themselves more vulnerable. I'm going to be doing that by creating home ranges of each of these herds' areas that they're living in using 51 GPS colored U's and their locations. And I'm going to overlay those movements in these home ranges that I've created and then create a habitat suitability model. So I'm going to look at where those escape terrain patches are and see where the U's are in relation to that. Another thing that might be impacting bighorn sheep habitat selection is cheatgrass. For those of you who don't know, cheatgrass is an invasive plant species that was brought over in the 1800s from Eurasia, so it's been here a really long time. It germinates early and competes very aggressively for soil nutrients and moisture, and because of that, it's sort of overtaken a lot of these native perennial plant species that we have. And a fun little fact about cheatgrass, the reason they call it cheatgrass is, to our knowledge, there's no real nutritional value to any ungulate species that eat it. So it's kind of just like you're cheating if you eat this grass. But the effect of cheatgrass on bighorn movement patterns has not really been looked into. So that's something that I'm hoping to accomplish with my thesis as well. So I'm going to be doing this. I've started by mounting a multispectral imaging camera on a drone and mapping the areas of Kamloops that have been most affected by cheatgrass. So that's sort of along Kamloops Lake and the South Thompson herds, those home ranges for those sheep there. There's a lot of cheatgrass. It really loves degraded habitat. So it pops up around there. So in the red here at the top left, the really dark red patches are the really heavy infestations of cheatgrass. And then the light red is, there's cheatgrass, but it's maybe like 20 to 40%. And those yellow dots are sheep locations. So you can actually see right now, before I've done any analysis, this was when we were first doing this, they're not really hanging out in those cheatgrass patches. So I'm going to be doing an analysis where I'm looking at ewe movement patterns throughout those cheatgrass patches and seeing if they're actually spending any time there. And then to further investigate cheatgrass, another question that hasn't really been answered is, do they even eat it? So I'm actually going to do a small feeding experiment at the BC Wildlife Park with the captive bighorn that they have there. And I'm going to be offering them a few different piles of grasses. One of them is going to be cheatgrass, and then a few other native grasses are going to be offered as well. I'm going to set up a camera in the enclosure, and over a 30-day period, I'm going to monitor which grasses that they're choosing to eat. And I'm really hoping that this helps address these possible factor contributing to this decline and also provide some really critical baseline knowledge on those relationships between cheatgrass and bighorn sheep. Thank you so much.
Okay, awesome. Yeah, thanks everyone. Yeah, as Nancy said, my name's Sean Seal, and I'm going to be talking a bit about forest fires and their impacts to aquatic ecosystems and also their impacts on fish food. Yeah, so obviously like wildfire is a bit of a hot topic right now, pardon the pun. And it's pretty easy to see as we drive around the province the impact that fires are having on our forests, on our communities, on the terrestrial landscape. But fires also have a pretty detrimental impact to our aquatic ecosystems as well. And these aquatic ecosystems not only provide important resources for our communities, but they also provide important habitat for very cultural and economically significant species like our Pacific salmon. So it's important to think about how aquatic ecosystems are also impacted by wildfire. The main way wildfire impacts aquatic ecosystems is by making the soil more hydrophobic and causing more water to run off the surface of the soil. So when this water runs off across the surface of the soil, it can pick up more particles of soil and sediment and also cause these to enter our rivers, which can have a few key impacts. The first of which is these big pulses of water and sediment entering the river can alter the channel structure and flow regime of a river. When this water picks up things like ash and burnt organic material, it can deposit that in the streams and also disrupt nutrient cycles, which we can sort of see here in Nicola Lake, can cause issues like toxic algal blooms and deoxygenation, which can result in fish kills, things like this. And then all of that sediment in the water also increases the turbidity or cloudiness of the water. And all of these things are potentially detrimental for not just the fish that live in these habitats, but also the tiny little invertebrates that they rely upon for food. So my research takes place here in the Deadman River watershed, pardon the wind noise, in Skichisinulu, the unceded traditional territories of Skichisin Indian Band. This watershed has been significantly impacted by wildfire the last few years, but most recently in 2021, the Sparks Lake wildfire burned over 50% of the watershed at a very high burn intensity. So as you can sort of see from the video that's played a few times now, that's what the headwaters of this watershed look like now. Very high burn severity, not a lot of pockets of vegetation left. And all this burnt habitat has been causing a bunch of soil to erode into this river system. And because of that, a lot of effort has been put into applying erosion mitigation treatments in this watershed to try to limit these sediment inputs. So our goal with this study that we're doing is to understand how wildfire affects these stream invertebrate communities. And by doing so, we can hopefully use the information we find to inform restoration efficacy and tie this work into other watershed values that have been identified as important to Skichisin, such as water quality and, of course, salmon populations, which we're all very concerned about these days. So question here is like maybe why macroinvertebrates? Why these tiny little bugs that live in the stream? Well, macroinvertebrates have actually been used as a bioassessment tool since the 1800s in Europe. And we have very well established relationships between certain taxa or groups of invertebrates and the sort of ecosystem parameters they need to really thrive and grow. So by identifying which groups of organisms are present, or sometimes more importantly, which groups of organisms are not present, we can tell a lot about the health of an ecosystem. Also, very importantly, these invertebrates are the primary food resource for a lot of freshwater fishes. Of course, salmon and trout are the ones we are thinking of most here. So what we're doing is we are collecting invertebrate samples using something called a Hess sampler, which is basically a big tube that goes down into the stream bed, and then the current flushes any bugs that might be present in that tube down into a net so we can examine them. We're also collecting invertebrates from the water column using something called drift nets, which are shown there over on the right. And these nets basically collect any invertebrates that have become trapped in the current or trapped on the surface of the water. And then we are looking in the lab and just sort of analyzing what invertebrates are present and getting estimates of what sort of biomass is there so we can get an idea of site-specific productivity. We've done this before and after these restoration treatments were applied. And it's important to sort of compare these two things because yes, it's great to know what animals are present in the stream bed, but by understanding how those animals are actually presenting themselves in the water column, that will inform the value of these different animals as prey to fish. Juvenile salmon are visual predators, so they rely on these invertebrates that have become suspended in the water column as a food resource. That's their primary food resource. They're not rooting around in the bottom like say a sucker or something like that. So the outcomes from this is that we hope to better understand how stream ecosystems respond to wildfire and use this information to more effectively target future stream restoration projects. And in doing this, we hope that we can help improve the quality of this watershed and better preserve our Pacific salmon stocks for future generations. So thank you.
This is Selena Carl, and she's going to tell us about, she's asking the question, where are the fish?
Okay, so hey, yes, my name is Selena Carl, and I'll be presenting my research titled, Where Are the Fish? I will be tracking juvenile Chinook salmon throughout the Okanagan River system with the use of, spoiler alert, acoustic telemetry. I know, crazy. Okay, so among the various salmon species, like Sean was telling us earlier, which are vital in ecological, cultural, and economic contexts in British Columbia, there exists a geographically and genetically distinct population of Chinook salmon in the Okanagan-Columbia River system. The Okanagan River is the only remaining accessible spawning area for the Columbia Basin Chinook population in Canada. So my study site is focused specifically on the southern Okanagan, flowing from OK Falls to Osoyoos, BC. And the Okanagan Valley is also the traditional and unceded territory of the Syilx Okanagan peoples, who lots of members were very, very helpful to me throughout my project, which I will get into a little bit in a second. But speaking of, the Okanagan Chinook is also known as Intiti, is one of the 4 food chiefs for the Syilx Okanagan peoples. Intiti historically had a population well over tens of thousands and provided for abundant food and trade for the Syilx Okanagan peoples. Unfortunately, though, anthropogenic impacts such as habitat loss, channelization, damming, and climate change have contributed to a rapid and detrimental population decline. These threats have resulted in the species being listed as endangered by COSEWIC in 2017. And according to multiple surveys conducted by the Okanagan Nation Alliance, which is the Okanagan Syilx Indigenous Government Group, they did a bunch of surveys in the fall of 2023. There was actually an estimate, which honestly was probably an overestimation, of only 20 adults spawning in the Okanagan River system, which is massive. Very, very detrimental. So in an attempt to fix this, the Okanagan Nation Alliance brought in a hatchery supplementation program in 2016, where juvenile Chinook are reared over winter and then released into the Okanagan River system come the following May. Unfortunately, though, there is a lack of data and information regarding the juveniles' habitat use and migration patterns post-release. So this is where my research comes in. So my research was proposed to aid in closing this data gap to optimize restoration and conservation. The main research questions are as follows. What specific habitats do juvenile Okanagan Chinook salmon utilize after release? And what is the outmigration timing of the juvenile Chinook? So I'm in the process of answering these questions. So over the summer, I collected extensive data on juvenile Chinook movements in the system. This was conducted with the usage of acoustic telemetry, which works by a tag transmitting signals, which are then detected and recorded by nearby acoustic receivers. So you can see, I don't know if I actually have a laser pointer, but in the bottom right and on the bottom left there, I had 2 types of receivers. I had a stationary one positioned in 15 different spots, and then I had a mobile receiver as well that I would use daily. So if anybody doesn't like needles, look away. But an acoustic tag was inserted via a hypodermic needle into the body cavity of 547 juvenile Chinook. A trained person did this. I did not do it. I don't like needles, so it wasn't fun. But so 2 days post-release, I released the juveniles into the system at 2 different release sites. So release location 1 is just below Skaha Lake and has been used as a traditional release site for the Okanagan Nation Alliance hatchery. So release site 2 is just below Vaso Lake within the restored naturalized section and was decided upon as a secondary to compare the juvenile survival between the 2 sites. So I would track the juveniles' movements via GPS and telemetry data, like I said, with my mobile receiver. With my mobile receiver unit daily in a specific area. I would also have my stationary receivers set up in specific locations throughout the system to record any possible detections for 24 hours a day. And I downloaded the stationary receiver data every 2 weeks. So, as with any project, some unexpected challenges came up. Over the summer fieldwork season, my mobile unit that was meant to track the tags with GPS components failed for about half the season, which was about 2.5 months. So that was really, really fun for me, and that resulted in my using several other pieces of equipment, which worked out okay. So I also had multiple instances of my stationary receivers being moved or completely pulled up out of the water by the general public, causing me to lose out on some of my data, and I had to redeploy them. This was indeed a challenge because some of them weigh close to 70 pounds. However, I definitely think the most strenuous challenge I had was downloading my stationary receiver data. So I would have to pull the receivers up from 2 to 3 meters below the water surface and bring them up onto the boat. This boat was actually made it a lot easier because normally we had a 12-foot tin can dinghy. And yeah, this involved a lot of heavy lifting and careful balancing so we wouldn't knock over, we wouldn't tip the little dinghy over. Had a couple close calls, but made it through. This was actually the last day that we had, so I was very happy to pull the last one in. So despite these challenges, I collected more than enough data, and I am currently analyzing the acoustic telemetry and GPS data I gathered over the summer. So through my research, I am aiming to fill in this knowledge gap of what types of habitats the juvenile Chinook use and their behavior post-release throughout this upcoming year. So this will hopefully aid in conservation efforts by identifying the important rearing habitats and bringing us a step closer into understanding the behavior of the Okanagan Chinook population and hopefully to bring our Chinook salmon back. So thank you.
Okay, so anyway, so Sylvie's going to tell us about what wildlife is worth.
Okay, so yes, my name is Sylvie, and my project is called What's Wildlife Worth? So this is a little something different. We're looking at the social side of the environmental sciences. I've been working with my supervisor, Dr. Peter Segaris from the Department of Economics, and this is a little bit about what I've been doing. So I want to introduce you to Finn, the burrowing owl, from the BC Wildlife Park. She was born in the park's breeding program for the endangered burrowing owls. She had a bit of a rough start in life. She was small and weak and had to be hand-raised in order to survive, but her early dependence on humans made her unfit for the wild. So for the past 8 years, she's been an educational ambassador for the park, featured in lots of education programs at the park and around the community. If you've ever been to an educational program at the park, or if we've come to you, you may have met Finn before. But now that you know a little bit about her and her story, what I want you to consider is, what is an animal like Finn worth to us? Would you consider financially contributing to her care? Also, what is the value of her role in public education? So these are the kinds of questions that I'm kind of looking into in my project. So I'm studying the BC Wildlife Park, which is our local nonprofit accredited zoological park. It's a recreational attraction for locals and tourists. It's a hub for field trips and education programs, and it operates conservation projects like a wildlife hospital and a burrowing owl breeding program, of course. So parks like this that have the focus on conservation and education have come a long way from things like colonial menageries and roadside zoos, you know, our tiger kings of the world. But these parks, the good parks, they often get a bad rap because of association with these more negative things and of past preconceptions. So these parks get kind of underrepresented and under-researched in terms of their social impact. But I believe there's a lot of valuable information there that hasn't been looked at very much. So that's what I'm looking for. But when we are studying these places, I don't think they should be considered just in terms of their market value, but rather the utility or the benefit they provide us. So that is what I'm studying. So these are my research questions. What is the recreational value of the BC Wildlife Park? What are guests willing to pay to increase educational opportunities at the park? And what are guests willing to pay to support burrowing owl conservation? So to address the first question, I'm using a method called the individual travel cost method, which is a economic method for assessing the recreational value of ecosystem services. And I'm applying that to the wildlife park. So it's a method for measuring the direct benefit enjoyed from visiting a recreational facility. So I've been surveying guests to find out their travel costs for a visit to the park and how often they visit. And the relationship between these two variables represents demand. This is a very simplified version. We also factor in things like socio-demographic information, which just adds a little extra confidence into the the relationship between the variables. And then what we're really interested in is something called consumer surplus. So we're going to find the averages here, and the area above the average cost is called consumer surplus, and it's the value beyond what the person actually pays for. And in economics, it's kind of considered to represent the happiness somebody gets from spending money on a service. So I have a couple of numbers to share so far. We're currently working on all of the math and figuring out the exact numbers, but these are some first findings from my surveys. My survey respondents found they've spent between about $13 and $529 on their visit to the park when you factor in all of the costs of travel, souvenirs they bought, things like that. And the number of visits range from 1 to 30 within a year. Also, don't worry about this, but if you know anything about statistics, this is one of our first little regressions that we ran. And basically what we're interested in is this tiny little number right here tells us that we did find a good relationship, a significant relationship between the cost of the visit and the number of times they visited. So we're hopefully going to get a good result. And I do have some extra numbers hot off the press, so new they didn't make it into the presentation. My supervisor's been crunching numbers, and he let me know that for a person from Kamloops, the consumer surplus that he calculated was about $25, and for those from, or no, that was from outside Kamloops, and those from Kamloops was about $68. So basically what that's telling us is that a visit to the park is worth a lot more to people than the $19 entry fee. So on to my other questions, looking at the value of conservation and education. For these, I'm using a method called the contingent valuation method, which looks at hypothetical scenarios for willingness to pay. And we're looking at 2 projects in the park's strategic plan, which are building a new education center and upgrading the burrowing owl facility. So we're asking guests if they would hypothetically be willing to donate money toward one of these projects. So we asked them, give them some options, ask them what they would donate. So some first things that have been found, I did 2 surveys, one in person and one online. So they were asked if one project had to be prioritized over the other, which one would they choose? In both of these surveys, they overwhelmingly chose the burrowing owl breeding site, so that gives us an idea of what people are interested in contributing money to. And then when it came to whether or not they would actually donate, we actually found a lot of people would, which was encouraging to hear. And interestingly, for the in-person survey, the majority of the respondents were not from Kamloops, so it was interesting to find out that they would actually be willing to donate money to a cause that was outside of where they come from. A lot of them were from outside Canada as well. And the amounts that they were willing to donate range from $5,000 to $1,000, with the majority falling between $25 and $50. So, in conclusion, what's the point of all this? So my research will hopefully reveal some of the value that a modern zoo can offer beyond just entertainment, quantified through guests' spending patterns presently and their willingness to pay for future projects. But rather than just helping the park increase revenue, I hope to kind of raise the awareness of the benefits that places like this can offer and influence some policy shifts toward external support and funding, as well as keeping the park accessible, so finding money from outside rather than just increasing costs for guests. And ultimately, I want to kind of help raise the social profile of facilities like this that are doing positive work, and kind of let people realize the benefit it can bring to their community. So the owls need all the help they can get. So thank you so much for listening to my presentation.
I have a question for the second speaker there. I live in Sun Rivers, and there's a number of ewes there that have collars on. Is that part of your study?
Yeah, it is. Yeah. I think I've got 9 sheep that are hanging around Sun Rivers right now. And then there's a few along the East Shoe Swamp Road, way down towards where the Lions Head is kind of where they stop. Yeah.
Anybody else? I have a question. question. So there's some of these populations are affected by disease, right? Not these.
Not
four.
the- no, not
four,
these
but
ones.
The ones in the Fraser.
The Fraser is really heavily impacted, and then the Okanagan bighorn sheep have actually just been wiped out by a disease called blue tongue, which is really unfortunate, but that is not a disease that we can really do anything about. There's mosquitoes that get brought up from the south, from some of the winds, and they carry those diseases, and it took about 95% of the population out. So that was really discouraging, but they're doing some preventative trials for another disease they've been struggling with up there, so we're really hopeful that that's going to help that population, because it's just, yeah, not doing so great.
Okay, anybody else got any? Yeah, go ahead. Go ahead.
And just while you've got the microphone, are there any plans for any kind of mitigation for this cheatgrass stuff, like way to subdue it?
Yeah, so one of the things I'm really hoping that when I sort of get my thesis out and we're looking at if cheatgrass impacts bighorn, then we can sort of move forward with the mitigation of it. In the States, there's been a lot of research done on what actually helps prevent the spread of cheatgrass. It's a really hard plant to kind of control. It really thrives in areas where other plants struggle, so it just kind of takes over. And it doesn't need a lot of nutrients. But from my understanding, they're trying some fertilizer, so you'll have a burned area or somewhere that's high risk of cheatgrass, and they'll put down this fertilizer, and it sort of suffocates the seeds, and it doesn't come through the fertilizer, but other native plants do. I couldn't speak too much on that, but I know there have been sort of methods that they're trying, because what they usually use is a very strong herbicide, but we don't really, that's not something we want to use, so.
Thank you.
Sorry, can I actually just speak to that? Sorry, I saw a presentation the other day exactly about this. But there's actually a pilot out of the Tranquille wetlands. And the difficult thing about cheatgrass is it stores a lot of energy in the rhizomes, like down in the roots. And basically, no matter what you do to smother it, it will spread out. It will spread its little tendrils and choke out anything near it. So what they've been doing is basically digging a trench around lots that they want to exterminate, and basically putting in little pieces of sheet metal down about a foot in the ground to stop the roots from spreading out, and then just throwing a tarp over it and smothering it, basically. They've been having some success with that, and then transplanting. They've been trying to few different species to transplant that will displace cheatgrass. So, anyways, I'm looking forward to it.
Okay, that's actually us. We're doing that. Green canary grass, not
Oh,
cheap grass.
sorry.
But it's bad grass.
Yes,
It's
my
bad
bad.
grass. Whatever. Anybody got any other questions? Yes.
I have a question for all the scat samples that were collected with the aid of the dog. You said you're doing DNA stuff to tell what they're eating, but is there any way you can do DNA analysis to see how many different fishers there are? Like, or is it the same guy, or is there 10 of them? Like,
Yeah, so this is actually really interesting. Fishers are notoriously hard to genotype and get DNA from, but I'm actually working with someone down in the States doing their PhD and providing them with samples to see if we can get fisher ID from the scats I've collected. So they're piloting that, and hopefully, like best case scenario, you could get a bunch of SNP panels and you'd be able to do lineage and stuff like that. But we're just piloting it to see if it works. So there is a possibility that we will be able to do that. But from my samples, we went to a lot of areas where we knew where fisher den trees were. So we're pretty confident about the scats being from specific fishers throughout the range, and they're far enough apart that we should have a good diversity of at least 6 fishers, possibly more, throughout that area.
Excellent question. Great answer. Anybody else? Julie?
The Okanagan salmon. So I've been seeing a lot in the news about incredibly high returns for some of the species, but it kind of sounds like it's not the ones you're studying.
So I'm uniquely qualified in that because I worked with Okanagan Nation Alliance for about 2 years, and I helped with a lot of their stock assessment crews, and the returns are the sockeye that are coming back. And that's why my project is, I'm not going to toot that anymore, but it's super important because the juveniles are really, we don't really know what's going on in our system, because in the States, they've been having a really, really big success on their Chinook returns, but for some reason in the Okanagan, they're just not coming back. And some suggestions are that the temperatures are just too high, and it's kind of creating that thermal barrier, and they aren't able to pass that. So kind of like us, salmon and fish just can't withstand certain temperatures. The oxygen concentration gets too low. So one of the suggestions was over the summer that thermal barrier kind of sets up on the American side of the Soyuz Lake, and they can't come back up. So the sockeye salmon have been doing really well, but for some reason the Chinook aren't. They could also possibly be just absolutely getting hammered for fishing in the States, because like 90% of the run is in the Columbia, which is in the States, or the Columbia, the United States portion of the Columbia. But we're not really sure, and I can't obviously comment on that, because I don't know 100%. But yeah, they've had a lot of success with the sockeye, but Chinook and sockeye are, despite being salmon, they're very different in behavior and kind of where they like to spawn and such.
So physical restoration is great, but it's not necessarily enough.
Yeah, exactly. That's why habitat restoration is so important. With sockeye, they're a little less, well, Chinook are very picky with where they like to spawn, and a lot of the habitats where they have spawned before have now just been completely decimated. I don't know if anybody's been to the Okanagan, but there used to be a really meandering river in the Okanagan, and now it's completely been channelized. Only about 10% of the original river is still there. So all that habitat has just been, is just gone. So that's one of the main parts, and that's why restoration is so important. But that's why mine is so, that's why my habitat portion of my project is crucial, because we need to figure out where the salmon are rearing so that we can kind of bring those habitats back.
Yeah, that's important. Anybody else got anything? Yes.
I have a question for the BC wildlife surveys that you're doing. Is there any research that, like as a general research, where people are asked, how much would you contribute or how much would you donate, and people fill like everything out in the questionnaire, but then is there any studies that compare that to how much they actually do donate or does get donated later on? Not just in your field, but a general kind of a?
Yeah, there are definitely studies where they do a follow-up. Unfortunately, that's not going to be part of mine. But yeah, when it comes to zoos, which is mainly what I've been looking into to see if other studies similar to mine exist, there's very few. I have not found any in North America, only in Asia. But some of them, I believe, did kind of follow up. I think what they did was then increase ticket prices and then looked if people still came. So yeah, and specifically related to what I'm doing, there has been very, very little. In general, when it comes to studying ecosystem services, I'm sure there have been more where they did compare, but I haven't looked super deep into that, so I don't have a really great answer for you on that.
Okay,
Hopefully that's
yeah,
what
I'm just curious.
Yeah.
Yeah, it's a tough question. Tough, yeah. So how about one more question? Here we go.
Sean, you have a slide of the micro bugs.
Yeah.
And there were 3, and the one in the middle looked like a rock. What was it?
Yeah,
That's why I left this on. You want to
it's
show us?
it's basically, that's just what we get in our nets when we collect these bugs.
Just a
Yeah, so if you guys,
Was
sometimes,
it off?
yeah, that was just a rock. There were thousands of little bugs in there, but basically that's what we're working with. That's kind of the tedious part. Yeah.
It's a rock.
Yeah. And there are just hundreds of little bugs lurking in all that algae. That's the really tedious part, is sitting in front of a microscope for 5, 6, 7, 8, 9 hours in a row and just picking out bugs. Is that a little bug? Is that just a stick? Yeah.
Imagine Brian Heisman spent his life doing
Yeah, so maybe it's in this store.
So this is a biter, right? When he bites you, and then a digestion.
Yeah, Bayless and Adelaide is the family. Yeah, that's a giant toe biter. I had one in my pool as a kid. It traumatized me a little.
And I used to live in a cattle pond in Texas, and in an inner tube, and they would come up and bite my butt.
Yeah, they'll eat baby turtles, baby frogs, baby fish if they can catch them. Yeah, they're pretty voracious predators.
Anybody? One more question.
Sorry, just one quick question.
Oh, two more questions. Okay.
You were studying the effect of like the ash and the detritus that whatever follows the forest fire going into the watershed area, right?
Yeah, it's being studied, not by me. That's kind of the hard part of this project I'm working on. There's a lot going into like studying these entire watershed scale processes. So I'm actually a small part of a much larger research team that consists of about 20 to 25 individuals. And there are geomorphologists specifically studying things like that. They have little traps in the river, and they're able to do basically a radioisotope analysis and identify exactly where sediment sources are. So based on just different elements present within the sediment samples, they can say, okay, this came from this drainage, this little port, like this percentage came from this drainage, and then target restoration.
Okay, and you're studying the effect it has on the little bugs.
Yeah, the little bugs to then see how productive the ecosystem is for fish.
And they're actually the big bugs, the
Yeah,
macro
the
invertebrates.
big bugs. The bugs bigger than a quarter of a million.
Jesse, did you have a question?
Yeah, for May, you said you had to bring a dog that was trained in scan detection up from the United States. Is there nobody locally or provincially that has trained a dog to do that? And should there be? Is there enough demand for that? Just because my uncle runs like...
I was going to say, and he's got a cadaver dog. Come on.
Yeah, I couldn't find anyone even in Canada. There's a couple, the road detection teams has a really long-standing history of being really good at what they do, and they're all scientists. They're not just people who like dogs and wanted to work with their dogs. They're scientists who started working with dogs, so they help with like the design and all of those other portions. But yeah, they also train people, and there's new companies starting up. If anyone wants to start a detection dog business in Canada, you have endless work.
Oh, that is so cool. That is so cool. Well, thank you so much, you guys. So let's thank our speakers again. See, it's yeah, it's amazing. I mean, science is amazing. It's full of so many people working together, each contributing a bit to a bigger story, and people who can translate it for the rest of us and tell us good stories are gems. So thank you so much, you guys. Thank you.